Hello and good day, friends. Welcome back to the channel. Always beautiful to have you here while we discuss our concepts in science. Today, we are just going to take a little time to discuss the concept of microbial culture, you know, using culture as a technique and why that is important in microbiology. Now, when it comes to the culturing of microorganisms in microbiology, we can consider that to be the cornerstone of what we do in microbiology. Very important that we are able to culture our organisms, and then whatever we culture from these organisms, we carry them through series of tests and analysis to be able to identify these organisms. And this is very important in what we do. When it comes to the culturing of organisms, have we take samples from all sorts of places. We take clinical samples, that's samples in hospitals, health centers, dispensaries. We can get samples from there. We take environmental samples, samples from soil, from water, from air. All of such samples we take. We can take clinical samples, we can take environmental samples, we can take soil samples, we can take samples from agriculture, samples from the field, from plants. So we take all of these samples, and it's important to us that from all these diverse samples, we are able to culture out different microorganisms for various reasons. Different kinds of microorganisms, some are pathogenic in nature because diseases. We want to be able to identify them. Some help agricultural produces. So for diverse reasons, we want to be able to culture out different microorganisms from different types of organisms. And this is very important in what we do. Now, when we get a sample, we usually prepare this sample, whatever the sample might be, whether it's a soil sample, water sample, fecal sample, food sample, whatever the sample is, we need to first prepare and process it. There are procedures that we take this sample through. Some of those procedures include you might have to do a serial dilution for the sample. So depending on if we look at the sample and based on our knowledge, based on literature, we feel that there might be quite a high number of microorganisms. We may decide to do serial dilution and then we serially dilute our sample and then culture or plate our sample. So we have different processes. Sometimes depending on if it is maybe a urine sample, maybe it's a fecal sample, we pass it through peptone water, we may pass it through other kinds of buffered waters and things like that. So we distilled water and other things like that. So we do all of this in order to prepare our samples. Now, when we finish preparing our samples, we now need to put our samples on what we call culture media or growth media. Now, these are chemical preparations that have been, prepared in the laboratory in very big companies that have the capacity to aid our microorganisms to grow. Microorganisms, they could grow on many different kinds of substrates. But we have special chemical preparations that were made that have been designed strictly for the process of helping the microorganism to grow. So there are so many of such media, and then the media are even in different kinds of classes. We have what we call general purpose media. These are media that will be able to grow any kind of organism, nutrient agar, your toothpaste, soy broth, but again, these are all what we refer to as general purpose media. We call them general purpose media because we believe that they are good for growing every or any kind of microorganism. Now we have what we call selective media. So these are media that aid in the growth of particular organisms to the detriment of others. So if you are targeting a particular organism, you could look for particular selective media which would help that organism to grow while impeding or making it very difficult for other microorganisms to grow on such a media. Maconke is such kind of media. Salmonella, Shigella, SSA, agar is another type. There are so many types of these media. You also have differential media. It's possible that you want to be able to differentiate. You have microorganisms in the same family that are similar. Maybe your enteric organisms, for an example. So you have your Salmonella, you have your E. coli, you have your Enterococcus. They are all similar, seem to be in the same family, and you want to be able to differentiate them. There are also kinds of media that you use, which we call differential media, which are able to help you differentiate one from another. And these are all different kinds. You also have the enrichment media, which are useful for the growth of particular specific organisms, most of the time pathogens. So these are all interesting medias that we have in microbiology. And now, when it comes to culturing, not only do we talk about media, we also talk about conditions. Important to note that there needs to be certain particular conditions before organisms are able to grow, even on the culture media. So there are incubation temperatures, and we have ranges. It is for organisms that can be found on human beings, organisms of human importance. Usually cultivate them between 34 and 37 in the lab. So, and if you notice, that is around the range of the human body temperature. So if the environmental sample 25 degrees is okay, 25 to 30 degrees is okay for them, then depending, if you take organisms as an example, like a sarcophagus, or organisms that we take from very cold and temperate regions, that we have to culture them under such temperature, under such conditions. So we tend to culture those organisms under maybe 4 to 10 degrees Celsius. So all of this is important. There's also pressure rating. Some organisms don't do so well if the pressure is high, while others do well. It's also important to note that there are different methods of cultivating the bacteria. You have the spread plate method, you have the stripping, you have the pour plate method. You also do membrane filtration techniques. If we're using something that is strictly water sample, for example, we can do membrane filtration, filter the water, and then the filter paper. The material that we use as a filter paper is what we put on the media to grow. We can strip using our inoculum. We could pour a little amount of the water. Now, I'll conclude by just looking at what are some of the advantages of doing a culture technique. Some of the advantages that we're able to recover viable organisms from a sample. When we have a sample, if there are organisms that are present and functional and living in that sample, we're able to recover these organisms from the sample. It's quite cost effective when you compare it with other methods of identifying microorganisms. Culture method quite cost effective and easy. You can also do phenotypic characterization of many of the organisms that you are able to get out from them. Now, the disadvantage of doing a culture technique is that culture technique is not able to pick up. There are some organisms that we refer to as non-culturable organisms. So if you do culture techniques for those organisms, you can't pick them up. It is difficult or impossible to pick them up. And some of them we don't even currently have any media used to culture such organisms. These are disadvantages, limitations, and it can be time consuming. It takes time to prepare the media, set the media, inoculate the media, wait for it to grow, and all of that. Lastly, we'll talk about where do we apply all these things, application of these culture techniques. We apply them in clinical settings many times. One reason or the other, samples have to be taken from human beings, different types of samples, blood, urine, feces, uh, different even tissues and cells can be taken and cultured. That's an application. In agriculture too, we do a lot of culturing of organisms to be able to determine the organisms that are causing one disease or the other. We also do environmental sampling. That brings us to end this video. I hope you learned one or two things. Remember to like, subscribe to our channel. You have a beautiful day.